The session today is on cell-to-cell communication and signal transduction. I chose to, do this topic largely because one, it was a newer concept that has been introduced in HL biology, and number two, the broad idea was to simplify the model of signal transduction pathway in a way that we can understand and apply in other knowledge forms as well. So basically, we are trying to address two questions here. One, how do cells communicate with each other without a voice, like we communicate? Do they have any specific mode of communication? How do cells respond to different signals that they receive? Now, cells communicate using a chemical signaling mechanism. And the molecule that is used in this communication is what is called a ligand. The ligand only binds to a specific receptors that can be found in the target cell. So this is really the one important way in which one cell can communicate with other cells in the body. Now, types of ligand. What are the different types of ligands that we have? So they fall into four major categories: neurotransmitters, acetylcholine, dopamine, serotonin. There are a number of these neurotransmitters that are found in the body. Hormones. Again, there are different kinds of hormones that are present in our body, and the whole lot of different kinds of chemical communications that are taking place. Ions. Calcium is one important ion that is used in a signaling mechanism. Cytokines. Cytokines are a group of molecules. Interferons and interleukins are examples of cytokines, and they form an important chemical signaling mechanism in the immune response system. Types of chemical signaling mechanism. This is more to do with the distance at which they communicate. So again, there are three kinds under this: endocrine, paracrine, and synaptic. Endocrine system is wherein it's more a hormone-based system, where there is a long-distance communication taking place. Insulin and sugar regulation are as an example of it. Pancreas, liver, and muscle cells could be involved in this signaling mechanism. Estrogen regulating the menstrual cycle is another example where estrogen is communicating with cells in the brain, the hypothalamus, and in the pituitary. So again, long distance communication there. The paracrine system is more local, not necessarily adjacent, but more local sort of the kind of impact that the molecule has. Cytokine is a very good example of it. The cells of the immune response system makes use of the signaling molecule to communicate with each other. Blood clotting factor is also another example of a paracrine system. Then we have a synaptic transmission. That is what you see in the neuron. The transmission, the information is moving across few nanometers in the synaptic cleft, but it is more, the distance is short, but it's more precise and faster communication. Now, what are the entire signal transduction pathway can be broken into three clear steps. Step number one is reception. Now, reception is the start of the entire signal transduction pathway, where a signaling molecule, which is a ligand, binds to the receptors that can be found on the target cells. The non-target cells are not going to respond because those receptors cannot be found over there. So the rest of the cells which don't have the receptors is not part of the communication. Step two is transduction. This is at the heart of the signal transduction pathway. This activates a chain reaction within the cell. This is sometimes called a signaling cascade. It is also referred to as the signal transduction pathway. Step three is response. The signaling mechanism ultimately results in the cells responding, and this response could be multiple things. It could be activating a gene, activating an enzyme, or it could be change in cellular activity. This picture puts the entire signal transduction pathway in a nutshell. Binding of the ligand to the receptor, which is called the reception. Step number 2, which is transduction, which is a series of chemical reaction. And the last, which is response, which is the response, the cellular response that is elicited from the signal transduction. Now coming down to reception. Now reception it all starts with the ligand binding to a specific receptor. What is important to know is the receptors are protein. Then the information or the properties of the protein will be helpful. We know that the proteins are made up of amino acids and they have a three-dimensional structure which are maintained by weak bonds. And that three-dimensional structure is extremely important in its function. Now, the receptors can be membrane bound or could be intracellular. Once the ligand binds to the receptor, what happens at the and there is a conformational change. Basically, there's a change in the three-dimensional structure of the protein. And that change in the three-dimensional structure of the protein is what causes the downstream signaling within the cell. And this is also responsible for the high specificity, that there is only one specific ligand that can bind with one specific receptor which can be found on the target cells. So it pretty much works on the same way as the lock and key of the enzymes and substrate. There are other differences between them. Much of the binding of the ligand to the receptor is a reversible process. Once the molecule dissociates, it just goes back to everything as it's just as usual. But the enzyme and substrate binding could result in an irreversible change in the substrate. This is much more quicker. The enzyme substrate complex formation is much more quicker, whereas a receptor binding, ligand binding to the receptor could, be for a couple of minutes to a couple of hours. So this actually picture kind of represents what a lock and key model is. Ligand is of a specific shape and size. It kind of fits into a certain domain of the receptor, which are found on the cell membrane. So this binding at the heart of it is a molecular give and take, and that association of that binding of the ligand to the receptor brings about a molecular change in the receptor. And then that change then has a rippling effect, right? It brings about further changes within the cell. So this is the model, so this is the model by which the entire signal transduction pathway works on. Now coming to the transduction, which is at the core of the process. Now transduction, as it's after the binding of the ligand to the receptor, the conformational change in the receptor can trigger a cascade of events or cascades of reaction, which could mean, in some cases, protein activation. In some other cases, it could be a protein enzyme activation. Sometimes there could also be formation of secondary messengers, such as the CAMP. The secondary messenger are important molecules that are formed in this transduction pathway, which could then trigger many reactions. And it's kind of could further amplify the process. So overall, transduction is an amplification process where a simple binding of the ligand to the receptor will bring about major changes in the cellular activity. Now, coming down to some specific examples, this what you see on your screen is where we are showing two neurons, one a presynaptic terminal and a postsynaptic terminal, right? So there are two cells, one adjacent to the other, and what has been shown in the orange is the presynaptic terminal, where the action potential has just arrived at the end of the presynaptic terminal, and that has caused vesicles containing neurotransmitters to undergo exocytosis. So they're migrating towards the end of the membrane. We know that migration requires calcium ions. Once the neurotransmitters are thrown into the into the synaptic cleft, these neurotransmitters will then go and bind to the the receptors that can be found on the postsynaptic knobs. So that binding of the neurotransmitter to the receptors on the postsynaptic cell will trigger the action potential. Now, how does that happen? To sort of explain that, we come to this picture kind of explains, kind of simplifies that model for you. So the circular spherical yellow structure is is a neurotransmitter, let's say acetylcholine. Now, this acetylcholine, which has been which has been expelled into the synaptic cleft, will now come and bind to certain receptors that are found on the postsynaptic cell. These receptors are cholinergic receptors, which are specific to acetylcholine. And the moment acetylcholine binds to these receptors, the protein channels to which the protein channels which are part of the receptor will undergo a conformational change. In its inactive form, it remains closed and it does not allow the ions to migrate. But once the ligand is bound to the receptor, that results in a conformational change in the protein. And that would result in these gated ion channels to open and sodium ions to migrate from the outside to the inside, turning the inner membrane more positive, depolarization to set in, and that triggers the action potential, which is then propagated along the length of the neuron. Now, here is another example, and this is the example of how insulin brings about a change. Now, you know that insulin is responsible for sugar regulation in the body. So the binding of this insulin to the receptor will result in glucose uptake. So here is an example of liver cells, what the effect of the binding of the insulin molecule to the receptors on the liver cells. Now, why do I say specifically liver cells? Because insulin receptors can be found in other cells, and depending on what kind of a cell it is, the metabolic transduction pathway could be different, and cellular response could also be different. So here is the example of what happens within the liver cell. So before we understand the signal transduction pathway, it is important to understand the structure of insulin receptors that can be found in the cell membrane. Now, the insulin receptors have two domains, one onto the outside, which is called the alpha domain, and then there is the beta domain. There are two units of beta domain to the inside. So when the ligand binds to the outside domain, which is the alpha domain, the entire protein receptor undergoes a change, and that change would result in autophosphorylation. Autophosphorylation is where units of inorganic phosphate would be added to the beta domain of the receptor, and these are added to the tyrosine residue. Tyrosine is an amino acid, so phosphates are added to the tyrosine residue, and there's a part of the beta domain which is called tyrosine kinase, and this tyrosine kinase will now have a catalytic function. And that phosphorylation of the tyrosine unit will lead to downstream signaling. What then there is a whole cascade that is set in place, and that cascade will finally lead to vesicles containing glucose transport proteins called GLUT4 to start migrating towards the cell membrane. So they move towards the cell membrane and they merge with the cell membrane, and the GLUT4 protein, transporter protein, gets embedded in the outer cell membrane. And these GLUT4 transporter protein then become channels through which glucose enter the cell through facilitated diffusion. And this is how glucose uptake increases, and glucose could either be converted into glycogen and stored, or could be used up is be transported to the mitochondria, where they could be used up to generate ATP. So this is one of the outcomes, possible outcomes of when insulin binds to insulin receptors found on the liver cells. Now we're coming down to another model of signal transduction pathway. In this case, the receptor involved is called G protein coupled receptor, and in short called GPCR. Now the GPCR operates in many different, cells and with different signaling molecules, but the overall model remains the same. So in this case, we are using the molecule epinephrine as the ligand. Now the GPCR has a domain to which the epinephrine can bind. Now the GPCR is a fairly long molecule which has remained suspended in the cell membrane. It's a beta pleated structure. It runs across 7 times. There is a loop that runs across the length of the cell membrane. But what is also important to understand is on the inner side of this receptor, there are 3 protein subunits, right, which forms a complex and remains attached to the receptor. Now, this protein complexes, one is called an alpha complex, the other the beta, and the third one the gamma. Okay. Now, these 3 complexes together form a complex and remain attached to the receptor. And in inactive form, there is also a molecule called GDP, which stands for guanosine diphosphate. It's a nucleotide, just like IADP, which is attached to the molecule, to the alpha subunit of the molecule. And that's the inactive state of the molecule. But when epinephrine binds to the GPCR, the entire complex dissociates and the alpha unit gives away the GDP and accepts a GTP, which is guanosine triphosphate. And this is now the activated form of alpha protein, which will then go on to activate a molecule called adenylyl cyclase. This protein is again a membrane protein. So it goes and binds to a certain site on the adenylyl cyclase and activates this molecule. The activation of this molecule is where it gets a catalytic function, basically again a conformational change in the protein, making it an enzyme now, which is able to break down ATP to CAMP, which is cyclic adenosine monophosphate, which is we call a secondary messenger, because it will then trigger a cascading, will have a cascading effect. So now coming down to what the CAMP does. Now CAMP, as I said, will trigger a signaling reaction, which is a series of reaction, which is called phosphorylation. So there is a series of phosphorylation which is going to happen. In molecular systems, phosphorylation is a common mechanism to make something which we have seen in other places as well, is a way to transfer energy to the molecule and make it more energy favorable. And also it's a way to, make changes, molecular changes in the molecule in a way that can trigger further processes. So there are 3 sets of phosphorylation which is going to happen. First with protein kinase, which is going to get phosphorylated, which becomes activated, and then that goes on to activate phosphorylase kinase. Then that gets activated, which will then again go on to activate another phosphorylase called glycogen phosphorylase. Now, the glycogen phosphorylase is an important enzyme which will then convert glycogen to glucose, right? And this glucose molecule will go through further small changes before it makes its way out of the system, right? And it's going to be from the cell, it's going to be released into the blood. Because epinephrine is a fight or flight hormone, right? And one of the things that needs to happen is that sugar needs to be made available to the cells for the sake of energy, apart from the other changes that are going to take place in the system. Now here is an example of another kind of signaling mechanism, and this time it is his testosterone is the ligand. Now, testosterone is a steroid hormone. Now, steroid hormones can enter the cell membrane. They can, they're small enough and they're hydrophobic, so they can make their way through the cell membrane, reach the inner cytosol, where they bind to specific receptors, which are the intracellular receptor. So there are these androgenic receptors to which these molecules can bind. And once they bind, they form a complex, and that complex will then migrate through the nuclear pore into the nucleus of the cell, where they get bound to the DNA, and that could activate many processes, right? Transcription and translation will follow, and that could lead to formation of muscles, growth, right? Muscle cells can form. It could lead to formation of semen, what you call sperms, or it could also lead to formation of secondary sexual, resulting in changes in secondary sexual characteristics. So that is an intracellular ligand here. Similarly, we have estrogen. Estrogen is also a steroid hormone, and this again also finds, has an intracellular receptor to which they bind. So they move in through the cell membrane, reach the receptor, bind to the receptor, and that binding, that complex will then move to the inside of the nucleus, where they could again trigger activating a gene, and that gene activation could lead to formation of different kind of protein, right? We know that estrogen is involved in the regulation of the menstrual cycle, particularly in the follicular stage, right? So if you look at how they regulate, we can understand a bit about how the entire communication actually works. In the initial stages of the follicular stage, where the follicles are maturing, there is a certain amount of estrogen that is released. And this low levels of estrogen will, suppress the receptors and will not allow the gonadotropin releasing hormone to release from the hypothalamus in the GnRH releasing neurons, right? And so therefore there is no further, that will not trigger the the formation of FSH and LH in the pituitary as well. But as the follicular stage moves further, as we get closer to the ovulation, there's a large amount of estrogen that is going to be released. Approximately about 24 to 48 hours before the ovulation, there's a surge in estrogen. So that surge in estrogen is what then is responsible to, which will go into the hypothalamus region. They enter the tissue, where they bind to the receptor and cause formation of gonadotropin releasing hormone, which in turn will then further trigger the formation of FSH and LH in the anterior pituitary. Right? So it's kind of a feedback mechanism. So initially it kind of suppresses it. So it's a negative feedback mechanism, but in the later follicular stage, it's a more positive feedback mechanism. And so there is a sharp surge in luteinizing hormone, and that really results in the ovulation process. So the overall working, though there's a fine regulation, and that regulation is working through using feedback mechanisms, but the overall signaling mechanism works on the same model, the ligand receptor model. This is again progesterone, which is also a steroid-based hormone, and then you can see that entering. So it enters the cell, binds to the receptor, and the complex goes into the nucleus, where it binds to the and activates a gene, and that gene activation could result in certain growth factors to form, and it's responsible for cell proliferation in the endometrium. Now, progesterone in the later stages of the menstrual cycle is responsible for maintaining the uterine wall in anticipation of a pregnancy, right? So one of the key functions of these hormone is cell proliferation, apart from the fine regulation of FSH and LH along with estrogen. Well, So I think that's the end of the transduction process. We come down to the last, which is responses. I've already spoken a bit about the responses while I was discussing the transduction pathway, but just coming down to some specifics here. The possible responses because of a signal transduction pathway could be gene activation, which could be gene switch off or switch on. It could also mean metabolic changes, which could mean glucose metabolism, protein metabolism, and fat metabolism, depending on what kind of cells we are looking at. It could also mean muscle contraction. The response could also be opening of ion channels, cell proliferation, cell changes in cell mobility and their migration, particularly during the embryonic stage, or even shape, right? So these are some of the responses that one can elicit from the signal transduction pathway. Well, we end it here. Thank you so much. The idea was to simplify it, and I hope I've been able to do that. I would be happy to get any kind of feedback on this, so that we can improve on this in our next session. Thank you very, very much. Thank you.